Good afternoon, everyone, and Eid Mubarak for those who are celebrating. We want to welcome Dr. Sharon Gerecht, who's joining us today from Duke University's Pratt School of Engineering, where as the Paul M. Gross Distinguished Professor of Biomedical Engineering, she and her students research how the microenvironment regulates cellular responses in healing and regeneration. And she focuses specifically on blood vessel physiology, aging, and disease. She's also the Dean of Research and Infrastructure in the Pratt School of Engineering at Duke, and a member of the Duke Cancer Institute. Dr. Gerecht completed her BA in biology at Technion, Israel Institute of Technology in 1994, and earned a master's in medical sciences at Tel Aviv University, and then a PhD back at Technion, before she moved to the US and completed a postdoc at MIT, and then joined the faculty at Johns Hopkins, and 2 years ago moved to Duke. So, in addition to that, she also has way more awards and honors that I can mention, but she is a member of the National Academy of Inventors, as well as the National Academy of Medicine, and multiple others. You can look her up online. But we want to give a warm Rowan welcome to Dr. Gerecht, and we look forward to your talk today.
Thank you so much for the introduction, and thank you for the invitation. It's always special for me to get invitation from students. So I 100% accept invitations from students. So thank you for that. I wanted to tell you a little bit about what we're doing in the lab, and everyone can hear me, I hope. Yep. So specifically about microenvironment regulation of vascular formation and regeneration. And I'll give you a few examples, but in general, our lab is interested in this. Okay, our lab is interested in understanding and controlling cell fate and morphogenesis. Specifically, we work with stem cells, either endothelial colony forming cells or pluripotent stem cells. We study how they differentiate into specialized cells, specifically the vasculature, and then eventually how they morph into a functioning tissue. Again, we focus on blood vessels. These blood vessels will grow and undergo homeostasis, which is the normal function of our body. They supply oxygen and blood throughout the body. But then at some occasions, we could have these extracellular stresses that change this homeostasis. And this could happen in different diseases. What we study in the lab are cancer and atherosclerosis. It does happen naturally with aging. It does happen with injuries, specifically wound, but it's also happening when we have exposure to extreme environment. And this is relatively a more recent years work we've been doing, funded by the DoD, where we have during aviation and space and different threats, we also have these extracellular stresses on the cell. So what we're trying to do basically is we're trying to understand how we can reactivate these processes of morphogenesis, so to be able to regenerate a healthy tissue and restore homeostasis. And in some cases also how we can even go further back to the differentiation and fate decisions. So we're trying to develop therapeutic, and even better, and this is basically what kind of the DoD would want to see, is more countermeasures. So we're expecting some kind of a threat or extreme environment, how we can prevent the injury, of course, to restore function, repair, and homeostasis. So we study many different stressors. One is the changes in the surrounding extracellular matrix, or ECM. We study low levels of oxygen, which are, it's called hypoxia. We also study changes in flow and in strain, and also more recently radiation, because it's important not just for a space, but also, of course, therapeutic radiation for cancer patients. So today I'm going to focus and give you examples relates to the extracellular matrix, and I'll see, hopefully I have time, I'll touch a little bit about, not a little bit, but I'll touch about hypoxia. So how we're doing that, one approach is actually to use engineered hydrogels to try and model the microenvironment. Why hydrogels? Because we can make them in two-dimensional surfaces or as a three-dimensional milieu. We can modulate and control the mechanical strength. We can make the hydrogel biocompatible, biodegradable. And kind of the most important thing to us is that we synthesize those hydrogels so we can basically define properties that are important for what we study. And indeed, using hydrogel for vascular engineering been done in at least the last decade. And there are some examples of how we can modulate the structure, is what I told you about the surface, also doing some patterning, different mechanics, adding different growth factors or precursors, and etc. So the composition. So with that, researchers managed to engineer blood vessels from arteries scale to capillaries and vein. And here's kind of a summary table of the different approaches they've been using, different compositions, cell types, and different factors. But our lab is really interested in understanding how things are happening. And for us, we wanted first to understand how the mechanics in the environment modulate the blood vessels. So this would help us to design materials better after we understand. So the first example I want to give you is about something that's called vascularity of the material, where basically is the ability of the tissue or the matrix to release stress when there is constant strain applied on it. So basically, this is from a nice review a few years ago, where it shows that most of the tissues in our body are viscoelastic. They have different levels of ability to release this stress, or to what we call stress relax. So when we started this study, there was starting to be some understanding that vascularity is very important for cell migration, but it was mainly studies in cancer cells, and it's been studies like a single cells. But we really wanted to understand how it's happening in the tissue level. So we developed those hydrogels, and I'm not going to go into too many details, but what you kind of need to know is that we have materials from the same composition. So both materials are made out of gelatin and dextran. So they have the same composition, but the way the cross-link is different. One is kind of more conventional, and the other one is more dynamic, where the dynamic allow the material to be more viscoelastic. So this was done by two postdocs in the lab. So Zhao is actually developed this material, and Rahel worked with the cell. But those of you that work with materials know that it's a lot of time to develop those materials, and then we actually hope that they won't kill the cell. So it's a lot of work, but we managed to create those hydrogel where we can make them soft or stiff, but within those stiffness to have two different types of stress relaxations. So we can take this hydrogel and actually decouple one aspect of mechanics, which is the stiffness, from the other aspect of mechanics, which is the vascularity. Okay, so you can see here we have soft material or stiff material, and then they have different properties in terms of the vascularity. So we said, what's going to happen if we'll take our cells, and in this case, we'll take the endothelial colony forming cells, encapsulate them in those hydrogel, and look if they're making vascular networks. So this is what we call vasculogenesis assay. would they make the same in the both hydrogels, or will vasculasticity impact? And what we have found that in the dynamic hydrogel, we have these beautiful vascular networks, and you can see them here more clearly. And hopefully you can see here, this is a Z-stack. So they have this lumen. This is like a hollow or luminized network, where we see much less, almost to none of that in the non-dynamic hydrogels. So we said, okay, now it's happening. We're glad that the vasculasticity is impacting, by why this is happening. What is the mechanism? So we know from other studies, both in vitro and in development of fish and mice, that vascular morphogenesis happening like that. So first of all, we have the cells, endothelial cells, endothelial progenitor cells. Then they develop this within the cells, they develop these vacuoles and small lumen. And then they branch, eventually forming this network. In vitro, when people are just using collagen or fibrin, this takes about 4 to 6 hours for the cells to make those vacuoles, about a day to start to sprout, and about a day and a half, 2 days to really make the network. And we said, where in this pathway or this process, the vasculasticity really matters. And we looked at the cells, and we saw that all the cells are having those vacuoles and beginning of lumen independent of the type of vasculasticity they're in. So we said, okay, this is not happening at this stage. It's happening something here when the cells start to sprout. So what we hypothesized here is that we have the cells in those hydrogels. So this would be the hydrogels network. And in the non-dynamic hydrogel or less viscoelastic, they just kind of stick around, they attach to the matrix, but they don't do much. But in the viscoelastic matrix, they are able actually to push away those matrix network. They allow them to push away and basically generate those sprouts. So we said, okay, let's see if this is really what's happening. So we did this traction force microscopy, where we encapsulated these little beads that are fluorescent, and we can look at them live, and we can see them moving much faster than in the dynamic hydrogel than in the non-dynamic hydrogel. Sorry, when we encapsulate them with the cells. So if the cells are moving around, these beads will move around too. And you can see here we quantify both the displacement, so how far they went and how fast, and we see that they're just moving more in the dynamic hydrogels. And they're doing that by basically contracting. And we prove that they are contracting when we stain for this phosphorylated myosin light chain. So the cells are able to contract and push those networks. What happens next is that this cell contractility allow for integrin clustering, and you can see here with stains of integrin in the dynamic versus the non-dynamic. We also did, besides quantifying that, we also find out in RT-PCR, and this leads basically for large focal adhesions, has been done by staining for vinoculin and the different quantification. But also when we try to inhibit that, so when we try to inhibit that the cells are stopping or not contracting, this is basically with this specific inhibitor with blabistatin. You can see here that we see almost no integrin, and then we see very little blood vessels to nothing. So it is that the cells are contracting, but it's not just that. What also happens is that because there is this focal adhesion, we would have phosphorylation of the focal adhesion kinases. And again, when we inhibit the phosphorylation, we see no vascular networks. This happens also with the cells actually degrading the matrix. So they don't just push away. So they're starting to push away the matrix, but it's not enough to make the network and touch each other. For that, they actually need to degrade the matrix. And this happens with metalloproteases here. And this is an example of a stain for the membrane-bound MMP. Again, when we inhibit these proteases, we don't get this network. And the cells not just digesting the hydrogel, they're actually depositing their own extracellular matrix. And you can see here with this PCR of a basement membrane of both collagen 4 and laminin in the dynamic hydrogel. And you can see this beautiful stain of collagen 4 surrounding a lumen of a vessel. So the cells really modify the matrix, right? So what we give them at the beginning, it's just the beginning, and then they're creating their own microenvironment. But those that creating this microenvironment allow this network to form. So kind of looping back onto why we're doing all of that, because eventually we want to create better therapeutics. So we tested that also in vivo. We took our hydrogel either with the cells or without the cells in the next one. And we injected them subcute to mice. And you can just look here. After 7 days, you can see these networks in the dynamic hydrogel, but not in the non-dynamic hydrogel. And we can see these various stains of integrin and focal adhesion kinases. So it is happening also in vivo. When we inject the cells, the hydrogel with no cells at all, we could also see that the dynamic hydrogels allow all these small blood vessels here in brown to penetrate into hydrogel, which is happening to much less extent in the non-dynamic hydrogel. Okay, so and this is when we injected a dye into the tail, we could see that the flow into those vessels is also happening. So these are functioning vessels. So this is all to say, if we're looking at this for therapeutic or designing or tissue engineered specific types of organs, we have to take into account the vasculoelasticity of the materials we're working with. So overall, what we think is happening is that those cells in the dynamic hydrogels are basically more contractile. They are able to push the hydrogel networks away. That allows for those integrins clustering, this focal adhesion kinases activation, degradation of the matrix, and deposition of new matrix, and eventually results in this robust morphogenesis. In a more recent work, Zhao started his own position, and he was very interested in what's happened, not just vasculoelasticity, but plasticity, which is another aspect of materials or another property of materials. So he first tested all these different organs. This is not human, this is rats. In terms of the plasticity, and we can see different levels of plasticity. I'm not going to get into details because this is really his work, but he managed to also do create those matrices that mimic those conditions or this plasticity, and he called it high plasticity, medium plasticity, and low plasticity, and series of studies. He has shown that it is actually impacting both vasculogenesis and also new blood vessel formation from none. So this is basically new blood vessel formation for existing, which is angiogenesis. So with that in mind, I told you about vasculoelasticity. Another aspect, of course, is the stiffness of the matrix. And there's a lot of work being done in terms of stiffness and cancer and even blood vessels. But what we kind of became interested in, this is Rahel really led this this work when she was in our lab, is really what's happening in aging. So no one can escape aging. But what we know in aging is that there's some reports that say that our organs become stiffer, right? And I think you can see that in the skin most importantly. And there's also, not necessarily related, issues with microcirculation of elderly people. We wanted to say, well, can we use maybe our in vitro system to try and understand that better? So the first thing she did, she just looked into, these are kidneys from old and young mice. So these are young mice and these are older mice. And she just stained them for blood vessels. And you can see the striking difference really just, in the kidney. These are relatively healthy, not relatively healthy mice. So they're just like 18 months old. She kept looking at them and looked into the collagen content. And we could see much more collagen in the aged kidneys. When she looked into collagen compared to cell numbers, we actually see less cells in those kidneys. So we have more collagen, we have less cells, we have less endothelium or less blood vessels. Okay, so what's happening to the tissue in terms of the stiffness? She measured the stiffness of those tissues, which is kind of an exhausting experiment, but she managed to do that. And she measured this through tissue rheology, but also atomic force microscopy. And it doesn't matter how she measured, she always finds that the aged kidneys are about twofold stiffer than the young ones. So she did show some correlation. So we see in the same kidneys, we see less vessels, we see more collagen, which results in stiffer tissue. So the question, is there really connection? Is the stiffness of the matrix really impacting the blood vessels? So for that, we wanted a material that where we can make and encapsulate the cells, make them blood vessels, and then make the tissue or the matrix around them stiffer. So make sure that we're not making the vessels in a stiffer environment, because we wanted really to have like, okay, this is our mimicking of the young blood vessels, and these are the blood vessels that are now exposed as a tissue, not as single cells, but as a tissue to a stiffer material. So she came up with this relatively simple system that was already done in the literature before in different ways, but where she basically encapsulates just collagen gels and hyaluronic acid, where each of them is metacrylated. So when we make these collagen gels in 37 degrees, they basically will polymerize just because of their physical structure. And then we can expose them to secondary cross-linker and link those methylacrylated. So this would be cross-link more than this one. So this would be stiffer than the other one. And indeed, she came up with this system where we can show that this is a not secondary cross-link and this is cross-link over time. We can make them stiffer. So we're very happy. We have that system. Can we do it now? Okay, can we make the network, expose them to the secondary cross-linker, which is, by the way, using visible light? What happened to that? And what she has found was very interesting, is when we have the control, so it's the softest one is 100 Pascal. We see this beautiful vascular networks here. They're staying with a V cadherin, so vascular endothelial cadherin, which is a marker for endothelial cells, and phalloidin. As the matrix becomes stiffer, you can appreciate that the phenotype is changing, right? We see less vessels and they don't look so great. And we can also quantify that. This is specifically the vessel length and the vessel volume. So it seems like it's happening what we've seen in that kidney. What we also see actually is that this vascular endothelial adherence seems to be starting to relocate away from the membrane of the cell. So we started not to see or to see less and less of this weak adherence. And it relates also to the beta-catenin, which is another correlation of this weak adherence. So you can see here that the beta-catenin in the soft are next to the membrane, but they're starting to move away from the membrane as the matrix is stiffer. Here too, she found that the cells are, again, they are under stress and they contract. So that seems like something they like to do. And again, by staining, in this case, she did staining, but also Western blot for phosphorylated myosin light chain. Here too, when she inhibited the contractility, she managed to recover some of the vessels. Okay, so here we see the stiffer, we have less vessels than the control, and when she inhibits contractility, there is less. So there is some level of rescue. And we could see also here that we rescue the weak adherent is going back to the membrane level in the inhibit compared to the non-inhibit, along with the beta-catenin. Continuing that again, the focal adhesion kinases are also activated. And again, when we inhibit those, we see recovery of the vessels. So basically, or rescue of the vessels. So basically what, yes, it's also happening in the weak adhering and the beta-catenin. So basically what is happening is that that with aging, we have the matrix around, at least in the kidney, but I would claim also with other organs becoming stiffer, and this is activating different signaling pathways that resulting in the adhering that are actually in the junctions of the cells start to move away from the membrane through this beta-catenin. So the beta-catenin move away from the membrane, and the weak adherent are basically destabilizing. And what happens then is that the vessels are not stable. And this is when they start to be leaky and dysfunction. So while we were working on this, we were so excited. You know, I showed you before, there is like, we compare 7 to 8 weeks to 18 months in lifetime of a PhD student. That's many months to wait for mice to get old. So what we said, well, you know, bioengineering can really maybe contribute and add this opportunity for using biomaterials to study aging. There's a lot of work that's being done more related to the bone, but we argue here that using really the biomaterials to mimic the aging niche. And, you know, we just looked in one aspect. Of course, there are so many aspects that's happening in aging, as well as more inflammation and everything. But really, there's a kind of an opportunity for us to understand that. So with that in mind, I do want to go and talk a little bit about what we're doing in terms of hypoxia. So why do we care about hypoxia? Hypoxia is actually happening or low percentage of oxygen during development, injury, and cancer. In fact, the cardiovascular system is the first to develop in the embryo. So it develops with really no active oxygen delivery. It's only passive oxygen. So hypoxia plays a major role. So hypoxia happens when we have oxygen consumption and limited oxygen diffusion. It activates signaling pathways of new blood vessels formation. And it's also the molecular mechanism is through this hypoxia-inducible factor and reactive oxygen species. And when we talk about hypoxia, typically we talk about 0.5 to 5% oxygen. And when we started these studies, more than a decade ago, there were limited studies on understanding how vascular differentiation and regeneration impacted by these levels of oxygen. And the studies are mainly, until today, using genetic modifications or gas chambers. Okay, so we take mice, we put them in hypoxia, or we knock up those hypoxia-inducible factors. And these are, most of the studies that were done in hypoxia are done for cancer, right? So we have the tumor grow, and it grows and grows, and then it needs more oxygen, and it's at some point sending signaling to blood vessels around to penetrate to the tumor, to feed it. But also, this is how the tumor eventually will metastasize, because from those vessels, tumor cells will travel throughout the body. So most of the study were genetic modification. They're typically being done in 2 dimensions, either in Petri dishes or more later years in microfluidics. And we have no real-time analysis of oxygen tension. And that's very important, specifically as bioengineers, because when you knock down a pathway, you knock down the entire pathway, but you're not really relating it to the environment around the cells. And you probably know that it's never black or white, right? When we have the tumor growth, it's not like hypoxic or non-hypoxic, right? We have the edges that would be less hypoxic, and we have the center that's going to be almost anoxic, almost with no oxygen. What about all the cells in between? There is a gradient of oxygen. How do they respond for that? So the first study that we've done with that was very simple. We just took our cells, or differentiated cells, pluripotent stem cells. This was done by graduate student Rowan. We differentiated them for 12 days, and we did that either in atmospheric conditions, so just in the incubator, 21%, or in what we call hypoxia, which is 5% oxygen. We try lower, the cells didn't survive. And just by doing that, what Savranti found is that our cells starting to do this in those, specifically in the hypoxic condition, they start to have these kind of colonies. And those of you that work with stem cells know that colonies mean that something is proliferating and growing. And these colonies are these endothelial cells, or vascular endothelial cadherin, V-cadherin positive cells, surrounded by a PDGF receptor beta cells, which are parasites-like cells. So the cells that will support blood vessels. When she continued to do that, what she found is that when she exposed the cells early on to low oxygen, so not later on, either we call it like secondary exposure or primed. When she get the prime, then she getting more cells, more endothelial cells than in the secondary. So that means that early on in differentiation, and whoever of you works with stem cells, early on in differentiation is really critical for cellular fate. So we've done that. We looked into the pathway. I'm not going to get into more detail into that. But then Bria joined the lab, and Bria really want to understand, okay, so we have those stem cells. Everyone is so excited about them. Their potential for therapeutics. But when they are going to go into our bodies, they're going to go into an injured tissue or a sick tissue, which most likely will have less vessels, right? If we want to regenerate. So she wanted really to understand how this hypoxia will regulate the integration of the cells that we're transplanting. So she did a really simple experiment first, and we used this for the retina, and I will explain why we chose retina. But she compared these iPS-derived endothelial cells to just mature retina. We can get that from patients. Mature retina, put them in hypoxia, she saw no difference. The only difference she saw is maybe in proliferation. We saw that the iPS derivatives proliferate better in the hypoxic condition. And then she took them and she moved them into 3D structures. And what she found was actually really stunning because she could see that in the hypoxic conditions, the IPS derivative makes these beautiful networks, which they did not do, which they did not make, we did not make in the mature cells. So we started to suspect that there is something with those IPS derivatives that respond better to hypoxia. And this is where I was still at Hopkins and I was collaborating with Elia Dow at the Wilmer Eye Institute. And reason being is because of retinopathy. So retinopathy happening could happen in diabetic patients where we have this healthy vasculature because of high glucose insult in the eye. Specifically, it would happen in the eye. We have some capillary dropout. As a result of that, there's going to be a lot of blood vessels growing in to compensate for that. And this results in what we call pathological neovascularization. If this goes untreated, patients can go blind. Okay, so that's a very important subject, one. Second, we have models actually to study that, and the model is oxygen-induced retinopathy. So what we do there, we take the mice, and they were just pups. On day 7, we move them to high oxygen level, right? So typically we have 21. We move them to a chamber of 75, and then immediately move them back to atmospheric conditions. This move basically creates this hypoxia shock. We said, can we inject cells right there and see if we can maybe normalize these vessels to prevent this pathological neovascularization? So we did that, and we injected these IPS derivatives compared to the mature retina, and what we could see that the IPS derivative integrates much better with the retina tissue. When we zoomed in on blood vessels, you can see here that the human IPS-derived endothelial cells are really integrating. These are the human cells, and this is the mouse blood vessels. They really integrate well, and you can see here kind of with the yellow-orange stain. But when we looked at the human retina, the mature cells, we could see the mouse vasculature, we could see the human vasculature, but what we've seen here that they're basically one on top of the other. So there is no real integration. They're both growing, but they're not really integrating. And we asked, well, why this is happening, right? And I told you that we have this hypoxia inducible factors. I'm not going to go into all of that. But what happened in hypoxia, these hypoxia inducible factors are accumulating in our cells. In normal condition, they will be degraded. But in hypoxia, they're accumulating, and then they're activating a lot of signaling pathways. One of the signaling is actually cell recruitment. And we said, well, maybe this is what's happening here with our system. So Bria went about and decided to look into SDF1 CXCR4 axis, which is the cell recruitment. So she took this hydrogel, just put inside this SDF1, which recruit endothelial cells, and put our cells on top of it. And I hope you can appreciate that the IPS derivatives were basically migrating into the hydrogel. So they were responding to the SDF1. When she looked into this, the receptor of the SDF1, CXCR4, she sees that in hypoxia, we have almost complete or almost 100% of the cells that in the IPS that are positive for this receptor. And indeed, when we inhibit this receptor, we could see that this is the control. We could see integration, but we see no integration when we inhibit this pathway. So what we think is basically happening is that when we inject these iPS-derived endothelial cells into this hypoxic retina, they proliferate a little bit better, but they definitely migrate better into these vessels and integrate them with them much more efficiently than the human retina endothelial cells. Now, I want to tell you one more story, a couple of others, but I want to tell you one more story here. So all of these studies were done basically in 2D. We take the Petri dish, we expose to hypoxia in those chambers, but you know, we love hydrogels. We wanted to move everything to 3D, okay? Because the vessels we're talking about is in 3D. And also maybe in 3D, we'll be able to follow these gradients I told you about the oxygen gradient in the tumor. Of course, we have oxygen gradients also for the immune cells. Our immune cells are flowing in the blood that is oxygenated, and then they leave the blood vessels into the tissue where there are lower levels of oxygen. So they are moving according to oxygen gradients. So can we generate, and that was basically my request for a postdoc that came to us several years ago, can we make those hydrogels that are hypoxic? Now, yeah, we can take any of the hydrogel I showed you and we can put them in hypoxic chamber, but we wanted something that we can do that on the bench, so we don't have to have the chamber one. And another thing is that we can look at it in live, in confocal, and see what's happening, really, how the cell's responding to oxygen. So Kion came up with this brilliant idea, actually, to generate these oxygen-consuming hydrogels. Not too many details, again, but we can modify, in this case, the gelatin with ferrolic acid. We can do the same for other polysaccharides. But by introducing this enzyme that called LECAs, we're creating this reaction that consumes oxygen. And it's consuming oxygen while it's making the network of the hydrogel. So imagine like it's on the bench, we have a polymer a solution, and then we're making the hydrogel. While we make the hydrogels, there is a consumption of oxygen. So we said again, can we make those, expose ourselves to hypoxia and see what happened in the formation of the networks? Now, I told you about the oxygen consumption by the polymers, but importantly, the cells consume oxygen all the time. So they're also adding to the hypoxic condition. So once the reaction is over, it's still hypoxic. And we can modulate that by modulating the concentration of the cells, by modulating the height or the thickness of the hydrogel. If you think about this diffusion of oxygen through that. Anyway, we managed to generate basically two systems of the same hydrogel where we have the cells in hypoxic conditions for almost 48 hours, and it's all, again, on the bench, versus a non-hypoxic hydrogel that they consume a little bit of oxygen at the beginning when we make the reaction, but then the levels of oxygen go up. So that was very exciting. And what we actually found is just the hypoxia versus non-hypoxia, no growth factor added, nothing. We could see these beautiful networks in the hypoxic hydrogels. We also show through different studies that what's happening here is, again, the cells are hypoxic, the hypoxia-inducible factors are stabilizing, and they actually induce these MMPs that degrade the matrix and allow the blood vessels to form. So that was very exciting because before this study, not all, but it was known that hypoxia impacting cancer cells that allow them basically to move. And we've shown that that's very important to blood vessels. So with that, I think I would like to finish. I'll show you that in a more recent work, we kind of wanted to understand better in terms of the gradients of these oxygen, and we can create this layer by layer hydrogels and control the levels of oxygen in each of them to the level that we can actually measure the different kind of distinguished levels of oxygen. And we did some RNA-seq, and we found all this interesting pathway. I'm not going to get into details because we're almost done, but all this pathway where we see that endothelial cells basically cluster together in hypoxia to save their lives. So if not, if they'll stay single, we will see that they'll upregulate reactive oxygen species and apoptosis. But once they're kind of aggregating together, they turn it around and upregulate cell survival pathways and cell cycle. They're making more connections and with each other, and eventually they can sprout into forming vascular networks. So with that, I'd like just to summarize that I showed you about matrix nature of dynamic in terms of stress relaxation and how it's regulate vascular assembly, dynamic collagen stiffening and aging in terms of the junction in the microvasculature, and how oxygen tension regulate vascular fate and integration. I didn't show you these two stories. They're too long and we don't have enough time. But I try to thank the people, that students and trainees that did the work throughout, but they're the ones that did all the work. Some of them are still in the lab, of course, our collaborators and funding. And I'll be happy to take any questions.
Beautiful.
Thank you.
In your last couple of slides, you were talking about the hypoxic gels and creating those using lacase as a consumer of oxygen. So two questions there. How long does the lacase work for? Like, what's the turnover number on that? And secondly, lacase is a copper-containing protein. Is there any concern about residual copper being left that could impact cell growth?
So I love the question because most people don't know lacase. So I really appreciate that. It's about 20, 30 minutes until the reaction is complete. And it depends on the modification we do to the matrix itself, right? So how many ferronic acid we add in there. So that's one thing. So if we leave the hydrogel, and I didn't show that because this it's already published, but if we leave it with no cells, basically the air oxygen will come in, it will diffuse in, and the levels of oxygen is going to go back up because the reaction completed. That's why when we encapsulate cells, they consume the oxygen and leave it low. Yes, there are different sources for lacase, and some of them kill our cells. It also depends on purification. But this specific one is from specific mushroom, works really well. And now, of course, Sigma stopped selling it. So now there's a collaborator in Mexico actually that grows them, and they do that actually for energy, the lacase, for energy studies, but they agreed to do that for us. So we're getting a blue powder from Mexico, but it's worked really well. Yeah.
In Rahel's work, did she look at anything other than kidney?
Tissues?
Yeah, any other tissues, any other organs?
No, she
in aging?
reserved them, but so those, I mean, it's amazing because I'm showing you this beautiful collagen. She had to do clearing of the entire tissue, and that like takes days.
Yeah.
And when she was done with the kidney, she was done.
Sure.
So she did reserve some. I mean, the other tissues are brain, which I don't think any of us want to really look into that. There is I think pancreas that are just reported in the literature in terms of aging and vascular and stiffness, but we did not do any.
Go on, students.
Do you ever see the converse in tissues? So are there diseases where there's an increased vasculogenesis? And then do you see a reduction of tissue matrix?
Now, that's an interesting question. No. Actually, that would be a good question for the retinopathy, right? That, but we have not seen that. I can tell you that I would expect that because to make the blood vessels, they need to degrade the mind. It needs to go somewhere. So I would think with that we'll have softer tissues, but no, we haven't looked into that.
I know in some stressful conditions, cells can donate, I guess, materials. I've heard it referred to as cellular altruism. And I'm wondering with your clustering that you see under hypoxia, if you ever see movement of mitochondria, lysosomes between the different cells to promote cell survival.
That's really interesting. We haven't looked into that. So we went directly to, first of all, the question was if we just put particles that are not cells, if they're going to happen the same, right? If it's really cell driven or just, you know, particle movement, but it doesn't happen. But we actually did not look into movement between the cells. Yeah, it's really nice.
Going back to Rahel's work again, when she increased the stiffness in the hydrogel while the cells were in there, you said she used visible light for the crosslinker,
Right.
for the cross, and that's as opposed to UV light, which I know a lot of crosslinkers use.
Right.
So that means with visible light, there wasn't as much ionizing radiation, so it's
Yes.
less due to the cells
Right. And I mean,
themselves.
she did check viability of the cells and all of that. Yeah, we wanted a reaction that's going to be, it's just with the UV, there were going to be too many, it's too toxic for the cells. We couldn't do that with the network already in place. So we had to find this kind of the visible light, which is not great because the cells grow in light, you know, so it's make it tricky, the experiment themselves, but overall that was the reason. Yeah.
Looked very convincing.
Yeah.
But
You had mentioned earlier about the timeline of sprouting and branching. Did you see that there was a specific viscoelasticity that you could have to align with that timeline, or like how the viscoelasticity could change how early on that sprouting occurred?
So that's a good question. That's one of the questions of the reviewers for the paper. So we have seen, so in the less viscoelastic material, is not that it's not happening, it's just happening really slow. And then when it's happening, the cell, you know, it's in vitro, so we have, you know, really defined time for the cells to make something or they'll die. So we are seeing that. So the cells will overcome to some extent because they can degrade the matrix, and I showed you they're making their own thing. They can make it work, but we can definitely try and harvest that to kind of control that process by modifying the material. Yeah. Okay.
more students. All right. Well, if there's no any more questions, I haven't seen any from online either. So if there's no more questions, then let's give Sharon another thankful round of applause.